Steve.
Where am I here? Oh, here I am.
There you are. Steve.
New bright light.
Okay, hi. Okay, so people are populating the audience. Okay, I think we can start. I would like to welcome everybody to the 21st and I would say the last talk of the Protein Electrostatics online edition. So this is the reason why I would like to make a few announcements before letting the speaker of today starting her presentation. The speaker of today is Lauren Webb. But before doing this, I would like to tell you a couple of things. Let me. Okay. Can you see my screen? Okay.
Yes.
So the first thing I would like to say is that our Protein Electrostatic YouTube channel actually reached the 100 subscribers, which was the minimum threshold needed in order to be able actually to put the name into the URL. So now we have officially the Protein Electrostatic YouTube channel, and I would like to keep it alive even afterwards and use this as much as possible as a repository for as a resource for people who would like to know a little bit more about the field or follow the activity of some researchers in this on these topics. This is also the reason why. So as I wrote to many of you, we had some regulation to follow in order to protect the privacy of people speaking and presenting at the conference. So I ask everybody to register through the site and give their consent to be filmed and to be recorded. And the original permission and consent was for 1 year. Since I think that given the feedback I've got, and I think that some of the presentation, all the presentation would be useful to be there for a longer time, I would send an email to all the speakers of this online edition, asking them to go on a link, to follow a link I will send in order to extend their consent to 5 years. Otherwise, in next year, I will be obliged to remove the recordings from the YouTube channel. So, of course, if you want them to be removed, there is no problem. But for those of you who are happy to keep their valuable contribution on the Protein Electrostatic website, I would ask you to give your consent to do this for a longer time. So this is one thing. Another thing is, as I told you today, this is the last talk of our webinar series. I have to thank everybody. It was a very interesting, exciting, and rewarding experience for me to manage this. I'm very happy and a bit sad that it's finishing, but actually it's not really finished for two reasons. One reason is that I'm actually so there was another meeting who was supposed to be there to occur in presence last year here in IIT, but that was not possible due to COVID. And this was a CCAM flagship workshop, which is again concerning electrostatics, but more on the computational side. This was organized by myself and Godard Hasutman from Jülich Supercomputing Center. And this is more related to the computational aspect and how can we exploit the new architecture, computational architecture, in order to extend our simulation to make them more effective, predictive, or being able to handle larger and larger systems. So I will send later an email to all of you with the details. For those of you who will be interested, this will again have a similar format as the protein electrostatic. It will be every Wednesday. Not this time, because this is a more European event, so it will probably be around 3 p.m. European Central Time. I'm sorry for people on the West Coast. It will be more difficult for you to attend it, but I had to. This is CCAM is a European institution, so I tried to find a trade-off, but it has to be more centered on the European time. And the last thing before starting with the talk of today is that I had from some of you some suggestions concerning people to invite on new talks. And so I think it would be a good idea not to close this in a definitive way, but I think it would be possible to keep it alive with, let's say, for example, once per month under the form of, I will call it protein electrostatic advances. Let's say 2 talks per time, once a month, to give room to the speakers who, for one reason or another, weren't able to speak in this first run of webinars. So I hope you would like this initiative, and I will send an email also concerning this. If you have suggestions, feel free to drop me an email and telling them to me. I'll try as much as I can to follow them. Okay, so I think I can start. I can stop talking, and I can start with the introduction to the speaker of today, who is Lauren Webb from University of Texas at Austin. So she's a professor of chemistry at the University of Texas at Austin. She got her PhD in chemistry in 2005 at California Institute of Technology. Then she did her graduate work in the laboratory of Nathan Lewis, where she studied chemical and electronic properties of functionalized silicon surfaces. Then until 2008, she was a postdoctoral fellow in the laboratory of Stephen Boxer at Stanford University. Her research focused on quantifying electrostatic fields in proteins using vibrational Stark effect spectroscopy. She then moved to University of Texas in Austin in 2008, Her research interests are centered on understanding and manipulating the mechanism of interaction, organization, and self-assembling of biological macromolecules in both natural and artificial environment. The title of today will be Experiments and Simulation of Electrostatic Fields Inside Proteins. Lauren, the podium is yours.
Okay, thanks, Walter. And I want to say thank you to you for keeping this organized. This has really been an enjoyable part of my week for the past many months. So thank you for your organization of this, keeping us all together and talking. So I gave Walter a very vague title and made it a little more specific here: Experiments and Simulations of Nitrile Probes to Measure Electrostatic Fields in Proteins. This is a topic of longstanding interest that I've shared with several of you before, and I'm going to give you kind of an update on work that we've been doing to really understand how we can combine spectroscopy of a vibrational probe in a protein with simulations to get a much better, I think more holistic and complete understanding of local electrostatic environment in a part of a protein of interest. And starting out, I'll just say, please interrupt me as we go on if you have questions. I don't think time will be an issue here. And I really want you to ask questions as we go. So before I start, I want to thank the people who've done the work. So this is now, unfortunately, a bit of an old picture of my group, but when we could all be together, and we have a couple new additions that have come on since then. The work that I'm going to be talking about today was done entirely by two recent graduates in my group, Jeremy First and Elisa Novelli. Elisa did all of the experiments that I'm going to be showing you, and Jeremy did all of the simulations. And I'd like to thank the people who've supported us, and all of the simulations that I showed you were done on our Texas Advanced Computing Center here at UT. So just by way of a brief introduction, as Walter mentioned, my laboratory is interested very broadly in what we consider to be electrostatic fields that control uncovalent, non-covalent biological interactions. So we're interested in understanding at a molecular level how these complex biological molecules can come together and interact in time and space in the really crowded and dynamic environment of a living cell in order to accomplish complicated function. And we study this from a variety of different perspectives. We're very interested in understanding electric fields at protein-protein interfaces. This is a topic which I've talked to this group about before. We're increasingly interested in what is going on in the non-covalent assembly of lipid bilayer membranes, and how electric fields that are created just by the structure of that two-dimensional environment then can organize heterogeneous domains within within membranes that have interesting function. And we like taking what we learned sort of over here from the real world and applying it to interactions that are artificial at what we call the bio-A-bio interface. So understanding how we can control biological structure and function through non-covalent interactions with materials such as surfaces, substrates, polymers, that kind of thing. And what I'm going to be talking about today is really much more fundamental than any of these given projects. It's going to be how we actually measure something as complicated as an electric field in a protein. And like I said, I'm going to be updating you on really what we understand about our measurement and how I think we can apply this now very generally, very broadly to a wide number of problems that we or that you or others in the field might be interested in. And, I know I'm preaching to the choir here, but I think it's always worth reminding ourselves just how complicated what we're trying to do here is, taking something, okay, Coulomb's law, very well understood, and applying it to these systems that have thousands of atoms in the protein and in the solvent. And, getting an electric field that goes from, say, this to this, and trying to understand how these highly local interactions then affect these large macromolecules inside these even larger dynamic systems like a living cell. And just figuring out what we're actually talking about here, figuring out what the magnitude of a field is, how far it extends, how far a change in field extends in a protein for allosteric effects, et cetera, is extremely challenging to both measure and predict, as we all know. And of course, for a long time, pKa has been a useful way to address this. So, you change protonation state of a molecule. And of course, one of those is stabilized versus another. And that change in free energy between that protonation and deprotonation event gives us a pKa. But you take that exact same molecule and you put it in something like a protein, which now has an electric field that's organized by the molecular structure of this giant macromolecule. And this can change distribution of, the charged versus a neutral state wildly compared to what our chemical intuition would be about happening in solution. And so these changes in free energy or changes in pKa have been a useful way to try to estimate what is going on with electric field inside protein for a long time now. But it's useful to remember, and I'm sure everyone in this community knows this better than I do, that when you change protonation state of something like a titratable residue, a side chain in a protein, you're also changing many other things, right? You're changing the number of possible hydrogen bonding interactions, you're changing its charge, you're changing its dipole moment. And so those are, of course, then going to have a responsive effect on the field that surrounds them. And so what we've been really interested in is trying to find alternatives to measuring electric fields in proteins that are perhaps less perturbative than something as dramatic as a protonation or deprotonation. And of course, what we're going to find as people who've studied pKa shifts know so well is that for any probe, it's going to turn out that our structural information is going to be crucial. And so experiment and simulation are going to have to go hand in hand in order to interpret experimental data. So our method of measuring electric fields in proteins is based on using the Stark or the electrochromic effect of vibrational oscillators. And Steve Boxer gave a beautiful talk on this. It was developed in his lab now, going on a couple decades ago. And there are now many people out here who are interested in using vibrational Stark effect spectroscopy to interrogate some sort of complex interaction or reaction environment inside a protein. I just want to make sure we're all on the same page about what exactly we're measuring. And so I'm just going to briefly review what Steve talked to us about several weeks ago now. And that is that if you take a harmonic oscillator that has a large difference in the dipole moment between ground and excited state, the so-called difference dipole moment, and you place that into a field, and that can be, say, an organized electric field inside a cell. It can be like an electrochemical cell, or it can be a reaction field of a solvent. It can be a field inside a protein, doesn't matter what. This difference dipole moment vector and the field vector will interact. And if that field is parallel to this difference dipole moment, well, that's going to stabilize both the vibrational ground and excited state, but it's going to stabilize the excited state more. And if you put it into a field that's anti-parallel to the dipole moment, it's going to destabilize both of these energy levels. But again, the excited state is destabilized more. And so that's going to lead in an increase in vibrational absorption energy. And so the experiment that we do is collect a vibrational absorption energy in some sort of background sample, and then begin to make perturbations to that affect electric field, and measure the change in vibrational absorption energy as a function of that perturbation. And that change in energy, change in the measured frequency, then tells us about the electric field change that has been caused by our perturbation, whatever that happens to be. Okay, so that's the basics of the measurement. And I showed you there the nitrile as an example. This is an entirely general effect, but the nitrile is in fact a probe that we really, really like to use. And again, Steve addressed some of its pros and also its cons several weeks ago. So benefits of a nitrile is it can be site-specifically placed inside a protein relatively easily. I'll show you how we do that in a second. It's small. It absorbs in essentially the only transparent region of a background protein vibrational spectrum. So it's easy to pick out a single nitrile from a large protein, just one nitrile in your sample. And it has a relatively large value of this so-called difference dipole moment, which means that it's sensitive to local field. And so small field changes are easy to measure with changes in absorption energy. But of course, it has some significant drawbacks as well. First of all, it has a large ground state dipole moment. And so, that's also a problem with pKa probes. And so putting it into a protein itself may cause a problem. That's something we've investigated and we can discuss another day. What I'm really going to be focusing on today is that it can accept hydrogen bonds through this nitrogen lone pair and through the pi bond around the triple bond. And this turns out to be a huge problem. It means really that any measured change in frequency in that very simplistic example that I showed you on the previous slide is actually a convolution both of a field measurement, a field effect that we want to make, but also a hydrogen bonding event that we don't necessarily know anything about. And so we've been spending a lot of time thinking about how to deconvolute this in our data. And I just want to emphasize this problem here. So this is a calculation done by Min Cho now several years ago, where he was calculating the frequency of the nitrile stretch in two different nitriles. So we have acetonitrile over here on the left, and we have methylthiocyanate over here on the right. The effect is the same, they just absorb at different frequencies. And what he's doing is calculating this frequency as a function of the angle that a hydrogen bond is making with the nitrile, okay? So whether it's interacting more or less with the nitrogen end of it, or whether it's interacting with the kind of pi cloud that surrounds this triple bond. And that turns out to be really critical because when a hydrogen is being shared with these lone pair electrons right here, this is a purely quantum mechanical effect. These electrons are in a pi anti-bonding orbital. And so when they're distributed a bit to the hydrogen, that strengthens the bond and it blue shifts or increases the absorption energy of the nitrile. Whereas if this angle is much smaller and interacting more with the pi cloud surrounding the triple bond, we're going to call these pi hydrogen bonds. Well, these are electrons that are in bonding orbitals. And so this is going to weaken the bond and thus redshift the absorption energy. And this effect is quite dramatic. These are very large changes in energy based on this angle. And so it's really just a way of driving home the fact that any structural change around our probe is going to, that changes hydrogen bonding, is going to affect our measured absorption energy also, and we better be able to quantify that. And in fact, I'd like to point out, we a couple years ago now, Cho's calculation has been out for several years, and maybe 2 or 3 years ago now, we did a study in GFP in which we actually were able to experimentally measure this. So what I'm showing you here on the right is previous work that I'm not going to discuss any further today, but we were able to put nitriles in a bunch of different places in GFP, and but just by sampling enough positions and then simulating those structures, we saw a distribution of hydrogen bonds from this sort of smaller angle that's more the pi hydrogen bond to the larger angle that's more the sigma hydrogen bond, and also saw this really nice correlation in absorption energy, exactly what Cho had theoretically predicted. So the work that I'm going to be talking to you about today was really motivated by two things. It was motivated, first of all, GFP is an interesting protein, certainly, but it's also a very strange protein. And so we were motivated by thinking about, is this generalizable, right? Can we do this in a protein that doesn't have a really convenient fluorescent chromophore that we can use as a handle, which was actually how we did this experiment in GFP. But it was also motivated by the fact that I have, one of the really enjoyable parts about working with this community and talking with this community so frequently is this model system SNase that's been developed by Bertrand Garcia Moreno that seemed like a really nice way of comparing a nitrile probe to a pKa probe. And I'll show you how we do that here in a second. I need to talk about one more experimental technique that we're going to use in order to try to deconvolute this field versus hydrogen bond effect. And this was inspired by some work that came out of Floyd Romsberg's lab now several years ago. And the idea behind this is that no matter what hydrogen bonding environment the nitrile probe is in, as you change the temperature, in fact, as you increase temperature, things are going to move a little bit more. Things are going to tumble a little bit more. Those hydrogen bond interactions are going to become, they're going to be less or have shorter residence times. I guess that's what I'm trying to say. And also there's a greater possibility that the pi cloud hydrogen bonding option is going to be possible because there's just more tumbling and sort of wiggling and jiggling, as Feynman would say. And so what we can do is collect vibrational adsorption spectra of the same probe, now not as a function of a perturbation, but as a function of temperature. Okay, and so why is this not working? And so what we're going to do is collect vibrational adsorption spectra over a range of temperatures at which the protein is stable, and measure the change in adsorption energy as a function of temperature. And that's going to give us what we call the frequency temperature line slope, which I might refer to as FTLS. It's basically just a slope that tells us about the sensitivity of that particular nitrile at that particular position, whatever it happens to be, to hydrogen bonding, okay? Where the greater this slope, the greater this perturbation, the greater the hydrogen bonding interaction is with the nitrile. Okay, and so as I mentioned, we turned to another model system to demonstrate this, staphylococcal nuclease, and again, inspired by the beautiful work that Bertrand's group has been working on for so long, that I'm sure many of you are familiar with, but just to make sure we're all on the same page, he's made many, many, many mutations, putting titratable residues all over this protein, interior, solvent exposed, et cetera, et cetera. And I'm just showing data sets for mutations, to glutamic acid here. And what you can see is that as you move this, this acid around the inside of the protein, you can get strong, many units worth of pKa perturbations in that measurement, okay, telling us that that is in a position of really perturbed electrostatic environment that is substantially changing the relative stability of the protonated versus the deprotonated state. But of course, we don't have to put just titratable residues there. We can put any residues there. And Bertrand actually already had a small library of mutations to cysteine. I'll tell you why cysteine is important here in a second. He was kind enough to share the DNA with, I think, 5 of those mutations. We made 5 more. And so the stars right here are the positions at which we mutated in a cysteine into the staphylococcal nuclease. And the reason we do that is, sorry, I'm having troubles with advancing the slides. The reason we do that is there's a very straightforward reaction for exposing a solvent-exposed cysteine to Ellman's reagent, this bis dithionnitrobenzoic acid. And under very mild conditions, this reacts to form this activated disulfide and a mole of side product, this TNB side product. And then we expose this to a strong nucleophile, leaving group goes away, and we're left with the nitrile on the cysteine and another mole of the TNB side product. And the reason this is so useful is that this TNB adsorbs strongly at 412 nanometers. And so a solution turns bright yellow. We can use the adsorbents to quantify the extent of the reaction and wait till we get quantitative yield of our nitrile side product. So in proteins that are easy to work with, like SNase, this is very straightforward. We could put the nitrile in many more positions. I'm showing you 10 because, as it turns out, this is going to show us a broad range of possible interactions between the nitrile and the interior of the protein, but we could do many more. We could do this in many other proteins, but this is just what we're going to stick with right now. Okay, and so what I'm showing you right here are the actual positions of these 10 mutations. And you can see, just like Bertrand had done for the pKa data, we sample many different possible environments of this nitrile. Some are buried, some are solvent exposed, and everything in between. And then what I'm showing you here on the right are just the raw normalized vibrational adsorption spectra of the nitrile at each of these 10 positions, okay? And then down here at the bottom, I'm showing you spectra of just the small molecule, methylthiocyanate, dissolved either in, aprotic DMSO or protic water. And the only thing I want you to take away from this series of spectra right here is that we see all kinds of different nitrile adsorption spectra. We see different energies, different peak energies. Where's my pointer? There it is. That's represented by this dashed line here. We see some that are even in a more polar environment than water itself, right? That's quite interesting. And we see very different line widths, okay? Something that we're going to quantify in what's called the full width at half max, or just the line width of the peak halfway up to the peak energy. And we see also very different shapes. Some are quite Gaussian, some have, very definitive shoulders. So we see a whole plethora of spectra right here. And apart from just being able to get these data, there's not a lot more I can say about that right now. There's not a lot more that we can do to interpret these data. One thing we did do, because we had all of the pKa data from the Garcia and Moreno lab, is we did compare these nitrile frequencies to pKa of the residues that he mutated in. So what I'm showing you right here is the nitrile frequencies for these 10 mutants that I just showed you the spectra of. And then on the y-axis is the measured pKa of, for example, the glutamate mutations at those same positions. So I'm just going to show you this for two, glutamate and lysine. And you can see there's really quite poor correlation between, say, a nitrile adsorption energy and a pKa of a titratable residue. They're measuring different things. We weren't expecting this to, be a beautiful one-to-one correlation, but it does emphasize that for every measurement we do, we really have to understand the details of what that actually means if we're going to interpret our data any further. Okay, so we were also doing simulations. I must have skipped over a slide. I'm sorry, I'm having trouble with my pointer, with my slide advancer. I skipped over this slide because this is just the strategy of what we're going to do in the first place. So I'm sorry if I didn't give you the signpost before. That was not my intention. So we have all these mutations. We're going to collect all the adsorption energies that I just showed you. And we're simultaneously going to do MD simulations in order to get a distribution of structures of that nitrile at each of these positions. So we get both the structures, and then once we have those structures, we're going to use the MD force field to measure the field at the midpoint of the nitrile bond, and then basically compare these data sets, and more than compare, see if we can actually use them together to learn more than we would from each one individually. It's not just testing, does the calculation match the experiment? Is if we have both of those pieces of information, what do we get out of all of that put together? A more complete picture. So I'm sorry for skipping over that. So I've showed you the experimental data. So these heat maps right now are then from our molecular dynamics simulations. And the way I'm representing those structures right here is as a function of this angle between the nitrile and an incoming hydrogen bond. Doesn't matter what the origin of that bond is. Could be water, could be an atom on the protein. And so,
excuse me, Lauren.
Yeah.
You have just structures of these things as a starting point for these simulations, or how are you doing that?
So we are starting with the wild type crystal structure. It's not wild type, actually. Bertrand made some mutations to stabilize. But we're starting with that crystal structure. We mutate in the nitrile. So we get all the atoms right. And then that's the starting point for the trajectory. We do not have crystal structures, NMR structures, anything of each starting point itself.
Thank you.
Mm-hmm. Okay, and so what I'm showing you, these data, I've just to kind of make this a little easier to look at, I've split up the data sets into hydrogen bonding from solvent and hydrogen bonding from an atom on the protein. And on the y-axis here, we have the range of this angle theta that we measured. So again, larger angle means more of a sigma hydrogen bond. And on the x-axis here, we just have distance. So either a very close hydrogen bond or something that's a little further away. And again, the only thing I really want you to take a look is that for these 10 mutants, these 10 positions that we've looked at, we see many, many different environments when we think specifically about hydrogen bonding. We see some nitriles that have a huge number of hydrogen bonds, sometimes to water, sometimes to protein. We see other mutations that have very few hydrogen bonds that we get from our structures. And then both the angle of those hydrogen bonds and the distance also varies. Some are close, some are further away. And so again, it's confirming what we see from our experimental spectra, that we just have a distribution of different sites here. So we really have a good way of asking ourselves, not what happens at one specific place, but what happens in general, right? We can see many different examples of what might occur. Can we learn something from all of this taken together? Okay, so something that I'm going to show you right here is keep in mind from the calculation that the Cho group did several years ago now, we are interested in trying to deconvolute hydrogen bonding events because the geometry of this is going to affect measured adsorption energy. And so this is our measured magnitude of this angle of the hydrogen bond versus our experimentally measured adsorption energy. And indeed, we see as this bond changes from our MD trajectories, so does the experimentally measured adsorption energy. So this is exactly confirming the problem that I laid out to you of how hydrogen bonding convolutes, our experimental data. What I've done here is the colors match. So, this goldenrod, for example, is T62, the nitrile at position T62. And what we've done is weighted each of these points as a function of how many hydrogen bonds. So something like T62, which has a lot, has a very heavy weight. Something like V23 has a very light weight. And then we've done a regression based on that weighted average. And we indeed see that there's a strong correlation between measured adsorption energy and this calculated angle, just like we were predicting in the first place. So indeed, we have a problem with interpreting our data if we can't go any further than this. But now we're going to do the second experiment I told you, where now we have a nitrile at a single position, and we're just going to measure it as a function of temperature. We do this from 5 degrees to 35 degrees, and SNase, as well as many proteins, are stable over that temperature range. So what I'm plotting right here is the change in the nitrile adsorption energy for all of these different positions of the nitrile, as well as DMSO, so our aprotic solvent here in kind of dark gray, and then water here in light gray. And again, what you see is that there's a wide variety of this measured slope. Okay, some are very shallow. They have very little hydrogen bonding interaction. Some are very steep. Okay, so again, we have a good sample of what an actual protein looks like here. What I'm showing on the right, what I'm showing on the right is this measured slope. Okay, so over here on the left, this is actual frequency, but now I take the slope of that, so change in frequency versus change in temperature, and I've just plotted that number versus the number of hydrogen bonds that we counted in our MD simulation, what I was showing you on the heat maps on the previous slide. And we see a very strong correlation between this measured slope and the number of hydrogen bonds that the MD tells us that is there. So indeed, this is a good experimental way of telling us how frequent and what kind of hydrogen bonds are nitrile seen at any given position. And so just an observation at this point, something that we should always think about when we use a nitrile in any context inside a protein is that it will have a significant amount of hydrogen bonding that is dependent on its exact chemical environment, that this will have substantial changes in vibrational absorption energy, but that this temperature dependent method is an in situ way of evaluating the extent of hydrogen bonding for any given position of the nitrile. So this is going to be a very useful experimental tool. And so the idea is, can we use this to actually compare to, benchmark, and understand electrostatics calculations? So the way that we're going to do this is quite simple. Like I said, we have our molecular dynamics simulations. We're going to put a point charge in the middle of the nitrile bond right here, and then measure the field in 1 step of MD at that point. For kind of historical reasons in my group, we tend to break this down into 2 quantities, what we call a solvent reaction field. Right here, the solvent reaction field, which is taking all the charges from the solvent and zeroing out the charges on the protein. And then the so-called Coulomb field, which is doing the opposite. It's zeroing out the solvent, taking the charges on the protein. And we're kind of in the habit of doing that again for historical reasons, which I'd be happy to talk about. But the point is that the field that we get from this is different and does not correlate with the absolute frequencies of the nitrile, right? So an absolute value of field from our calculation does not correlate with an absolute value of the nitrile absorption energy. And on the face of it, that may seem disappointing, but then the question we ask is, well, we're doing a difference experiment, did differences correlate? Okay, so what I'm showing you here is now, again, for our 10 mutants, our 10 positions of the nitrile, I am showing you the distribution of fields that we calculate from our distribution of structures from the MD trajectory. That's just binned here, these kind of gray histograms. And I'm showing it both as a function of the field generated by the solvent and the field generated by the protein. Just to kind of break up these data sets. And I'm going to put next to it our experimentally measured vibrational absorption spectra. And just qualitatively by eye, you can see, there might actually be some similarities here, right? So V66, for example, was notable in our experimental data as being a very narrow peak. We get a very narrow distribution of fields. Whereas this, say, N118 position was notable for being a very broad peak, and actually we get a broad distribution of fields. So qualitatively, in this kind of hand-wavy sense, there appears to be some replication of our experimental data here. Can we go further? I've broken these up again by whether the field is from the solvent or from the protein. And if we then we look for one number, and we've just picked standard deviation of that field over the course of the trajectory, and we plot that standard deviation as a function of the line width. So what we call full width at half max, which is literally just the width of the vibrational absorption spectrum. And again, this is an experimentally measured quantity. We get, okay, correlation. It's not quite clear what that means in a real system where, of course, every charge is contributing to field. And so if we take the standard deviation of these 2 calculations put together and plot that against full width at half max, we now have a very good correlation between, again, the line width predicted by the simulation versus the line width that we actually measure. Okay. And so here's the final step. So like I said, just qualitatively by eye, you can say, well, okay, even though we're not getting absolute values of field correctly, we are getting differences, we are getting distributions, and that's quite exciting. So I'm going to take this just a little bit further. And this is in the spirit of let's use experiment and theory together, not just compare them one or another, but actually use one to enhance the other. So these are, again, our 10 mutants. This is the total external field just binned in these histograms, like I've told you. It's in kBt over E angstroms. And so we're going to just do the energy conversion to put it in units of wave numbers, like our experimental data. And then because the absolute value of the x-axis was wrong, we're just going to scale the absolute value of wave numbers to our experimental data. So we're going to match the height of our calculated distribution to the maximum energy of our experimental peak here in the dashed line. Okay, so this is essentially the same thing as the side-by-side comparison I was showing you, where you just kind of eyeball, okay, wide, okay, that's great. Now, by doing this energy conversion, I can actually superimpose the experimental data in the dashed line over the simulation data in the gray bin. Okay. And so here's what we're going to do. Remember that our FTLS is this temperature dependent experiment, and that is a completely orthogonal measurement of the full width at half max at a single temperature. Okay. They're well correlated, as I showed you before. This is the same data I showed you a couple of slides ago. They're well correlated, but they're different, okay? And so what I'm gonna do is I'm going to take my simulated spectrum, and I'm gonna scale the full width at half max by the quantity of the FTLS. So I'm essentially going to change what's here on the x-axis by what I know about here on the y-axis. And we did this by taking our two extreme values. So we took our smallest FTLX, the V66 here in red, and we took our largest N118 here in this dark blue, and we made those two endpoints match as well as we could. And then we scaled the bandwidth of every other distribution based on the magnitude of the FTLS. Okay, so here's the data that I just showed you, this superimposing experiment and theory. And here's what happens after we do this scaling. So the dashed line is still the experimental data, and the colored line is now the spectrum that we are predicting based on the distribution from the simulation plus the scaling factor from the FTLS. And what you can see, and keep in mind, there's a star next to V66 and N118 because these are our two endpoints. These are the ones we tried to fit as well as possible. Everything else is just based on the FTLS. And what you can see is now for many of our experimentally measured positions, the prediction of experiment and theory are actually quite good. They're good in getting the shape of the spectrum. They're good in getting the line width. They show us very clearly, for example, where a shoulder might emerge, for example, that we would see as structure in our vibrational absorption spectrum. And so we're very excited by this because this is essentially a way of now approaching a new system, whether it's a new protein, a new position of the nitrile in a protein that we've already studied, et cetera, and making a prediction based on the MD simulation that actually, through really straightforward calculation steps, gives us a prediction of the experimental spectrum. And so we can now use this to predict new and interesting places to put the nitrile that then we then go and do experimentally in the lab. That seems to be highly generalizable, at least for a protein like this that is easy to work with. Okay, and so with that, I'm just going to put up my conclusions. There's plenty of time for questions. So what I've been showing you is that electrostatic information is contained in vibrational line shape, yes, but it needs to be deconvoluted from hydrogen bonding that's occurring. And that we believe that in particular, this temperature dependent spectroscopy method is a very efficient way to do this that is complementary to all of the other spectroscopy that we've been doing, and that then can be combined with MD simulations to tell us much more about the individual molecular environment surrounding a position of interest that we're studying with our nitrile probe. So with that, I'm going to stop. I see there's some, yeah, everyone's thanking Walter. Excellent. I don't see any questions for me yet, but please.
Of course, that will be. So I see already Thomas raising his hand. Thomas Simonson.
Okay, thanks, Lauren. It was very interesting. You showed a lot of data. I don't know if I, understood everything. I think on just the previous slide, what I understood is you have a lot of cases where you have a really nice match between the kind of the MD fluctuations and the measured widths, and then you have a few sites where there's poor agreement, like site 118. I may have missed something, but I was wondering how you interpret the poor agreement of a few sites. I mean, could it be due to, say, limited sampling with the MD, or could it be due to sites that are more sensitive to the water model, which is perhaps not so good? This slide, there was a previous slide.
Maybe this. I mean, this is showing sort of everything that I was trying to show in the side by side, which is a lot harder to work at, to look at. And I think what you're pointing out, let's look at N118. So the black dashed line is the experimental spectrum, and the gray histogram is the prediction from experiment. So I think that's what you're highlighting, right? And yes, in just the straight up out of MD, it is broad, right? But it's not nearly as broad as the experimental spectrum. So that implies to me that we haven't adequately sampled all of the positions of the nitrile, right? And so is that because we need to do more MD? Is that because our water model isn't as good as it should be? Probably all of the above. And I guess what I find so exciting is that even within that limitation, because we have experimental data to scale our simulation against, we can still get fairly good overlap between our final prediction and our experiment.
And then one other detail, but it's somewhat important. I think it's very exciting now that the quality of simulations is kind of getting to the point where they can indeed help do the things you're doing. And it's already nice that with these, I believe these are fixed charge simulations, if I understood correctly. But I think probably maybe you have plans to go to more sophisticated force fields, and we already know they do actually quite a lot better for some properties.
Oh, absolutely.
The same department as Pengyu Ren, I didn't realize that. So
yeah, absolutely. So so yes to everything you said. So these are done in amber. So yes, fixed charge. I should have. Oh, I don't know if you can all see that. Every Wednesday at 11:30, there's an emergency test from the university. It just flashed up on my screen. Sorry if you saw that. Fixed charge, blah, blah, blah. And so, yes, we are collaborating with Pengyu on Amoeba. And we're doing that. I mentioned very briefly, we've also done a lot of kind of model system work on GFP, which is a really nice system to work with, just kind of a sandbox. And so we're working to do simulations with Amoeba on that. And the question we're asking, so I guess the way that I think about it is, it's not just, does the more sophisticated force field give you a better answer? It's what are you actually looking for, right? Is it going to be worth the extra effort, the extra computational firepower, et cetera? Or is a simple model actually good enough, right? So we originally got into this these many years ago, thinking about differences between, say, a continuum versus, a point charge type of calculation, right? Well, the answer is it really depends on what you're looking for. It depends on the level of accuracy that you need. And so I'm really excited working with Pengyu to address exactly that question, right? Do we get information that actually helps us interpret what's going on inside the protein better, or do we just get better distributions?
Thanks, Larry.
Adrian?
Yes. Hi, Lauren. That was
Hi,
great.
Adrian. Thank you.
So I have a question. I'm of the family of people that says that most of Bertrand's results cannot really be ever interpreted on a single conformation, right? That really what's going on with these mutants is that either you open a channel and the water comes in, or or you get a conformational change that when you protonate the residues, you protonate the lysine or you unprotonate the aspartic, they find a way to be solvent exposed, right? That they're not really in the core. So in that sense, I'm not fully surprised that your results don't do the pKa, because kind of I think they shouldn't. But the question is, if you do something like try to unfold your system with either urea or higher temperature or something, do you see a shift towards a fully solvent exposed nitrile?
Yeah. So what happens when it's, say, fully unfolded?
Yeah.
Is that, yeah.
Right.
Absolutely. And people have been using nitriles to do that for quite some time.
Okay.
Fengai, for example.
Right.
Putting nitriles in different positions of a protein, essentially to ask what's the level of secondary or tertiary structure at this particular position, based on whether it looks like it's mostly in water or mostly not.
Okay. And the other one is that I think one experiment, I think one needs to think about this very carefully, but you have, is what if you take one of Bertrand's mutants, right? The ones that are really shifted, and you put your probe next to it, right? Or in the neighborhood to ask the question, how is the electrostatics around that residue changing while you titrate with pH? There's probably complications, but that to me will be very interesting, right?
Yeah, I completely agree.
Okay. Yeah. So next time we talk here is done.
Next summer it'll be done.
Okay, cool. Excellent. Thank you.
Thanks, Adrian.
Okay, then Chu Zhang.
Hello. Thanks for the great talk. So I'm just wondering. I think you have already mentioned it briefly. Could you elaborate more on the physical origin of thermochromosome? There's more jiggling and wiggling, like as you increase the temperature, but how does that relate? Does that mean that the probe experiences a larger field, or like there's more hydrogen bonding? And I'm also wondering, have you done something like temperature dependent MD simulations, like you change the temperature and see, let's say in water, and when you change the temperature, how does that change the environment around the probe?
Okay, so to address your first question, I think the way I visualize this goes back to this picture again from Min Cho, which is that the change in nitrile adsorption energy that we measure as a function of temperature is coming about because of all of the different ways that a hydrogen bond is orienting itself around this nitrile. And at higher temperature, where you have a little bit more thermal motion, there are going to be more possible ways to do that, with the wiggling and jiggling, right? And so anything that has a strongly hydrogen bound interaction at whatever your base temperature is, let's say 5 degrees, that's going to weaken just because there's more fluctuation as you go to higher and higher temperatures. Right.
Yeah, so actually, yeah, that's right. Actually, I forgot, when you increase temperature, does it redshift or blue shift?
It redshifts.
It
redshifts. Yeah, redshifts. I don't know if I can find. So this is just a schematic, but it's showing a redshift. Which is what we observed for every experiment we've done thus far. You asked another question about temperature dependent on molecular dynamic simulations, and no, we have not done that. So an interesting question that we could ask, I won't scroll back to that final slide, but would our distributions get better right at the outset if we actually were collecting a distribution for every temperature that we were measuring? our FTLS data at, and we simply haven't done that.
Oh, and also I guess because you already showed the data like DMSO versus water. In DMSO, the slope is much smaller, and I think that makes sense because there's I guess less hydrogen bond. So I guess probably in a solvent where there's absolutely no hydrogen bond, let's say hexene or something, probably there's basically the slope is basically 0, I guess.
Yep, absolutely. And I won't try to scroll to it. We've done this in, I don't know if we've ever done hexanes, but THF, for example, it's another good solvent to work with. Yeah, and the slope is very, very shallow because there's essentially no hydrogen bonding.
Okay, cool. Thank you.
Okay, Marilyn?
Nice, very nice. Great to see you. So the question, I have 2 questions. I mean, one is this, can I view this as at least a partial validation of getting hydrogen bonds out of MD generally? I mean, I don't really care about nitriles, but I do care about, waters and side chains. And so do you think that to the degree that this works for you, that it's just generally going to work?
So I can only tell you for nitriles because we have a very nice experimental way of testing it, but it has worked very well. Yes.
And so if you can kind of squidgy that to make the simulations work a little better, then hopefully that will, I guess the question is there some way you can use other probes or think about other things that would make this a little more generalizable? But I think it's nice. The other is, I mean, I'm really into these protonation microstates these days. And so, even in staph nuclease, you're going to have a distribution of protonation states in your equilibrium mixture. And, I'm wondering whether maybe the broader ones might be near protonatable sites or near sites with a larger, more microstates with different charge in the vicinity.
Yes,
even if they're on the surface, you're not looking at huge changes in field, but if they're fluctuating, you'll see that.
Yeah, that's a good question. And from the data that we have so far, for example, we could look at that maybe with the MD simulations. Where's my pointer? Right, because we definitely see. Sometimes we have a broad distribution of distances. Sometimes we have a broad distribution of angles, right? And then going in, the only thing I'm asking here is just counting hydrogen bonds, but then going in and looking at the chemical details of each of these structures and of the different representative examples that we see based on the different types of simulated hydrogen bonding interactions. I think that's exactly what you're asking about. And certainly for, keeping in mind that we're really using SNase here kind of as a sandbox, we certainly do that in other experiments where we're asking specific questions about, say, a chemical interaction, maybe across a protein-protein interface. So I think that's exactly where this goes then. It points you in the direction of why one environment is different than another.
Well, what I'm actually thinking of is something that you can't see in the MD, which is that in your test tube, the protonation, you don't have all one protonation state. You have a distribution of protonation states as well as a distribution of ionization of structure state. So I'm going to start saying, thinking you have one protonation state in your test tube is like thinking you have a single rigid structure. That's just not right.
Yeah.
And
okay.
So we can look at the microstate distribution for SNase and see whether different regions are showing up with residues that say are more likely to be changing charge in the low energy microstates.
Yeah, I see what you're saying now, and I agree with you completely.
Okay, it's a thought. We can talk.
Yeah.
Okay, Yasmin. Yeah.
Hi, Lauren. Thank you for a really interesting talk. I have a couple of questions about the MD part of your experiments. And the first one I was curious about is sort of a follow-up of Steve's question, because you start from a homology model, basically. How long do you actually sample to get these results? And do you allow the protein to sort of be relaxed before you start sampling?
So yes, right. So we mutate in our nitrile. We allow it, we correct temperature and pressure and allow it to equilibrate and then start the trajectory. I should have mentioned that we're using an umbrella sampling strategy here to try to get a Boltzmann weighted essentially distribution of the thiocyanate, because actually I'm glad this slide is up right here. Unlike some amino acid side chains, we have some problems here because this can freely rotate around both its chi2 and its chi1 angle. And that's going to lead to really substantial, not just hydrogen bonds, which I've sort of reduced to right here, but actually just where the nitrile is pointing to begin with. And so we use an umbrella sampling strategy with a high energy potential around a range of both chi1 and chi2 in our trajectories. And so because of that, I want to say the simulation length for any given trajectory is fairly short. I'm thinking maybe 50 nanoseconds or something like that. And we could do much longer on TAC. But the point is that we have many, many trajectories at many different starting angles of the nitrile and then binning all the data through umbrella sampling.
I see. So in your umbrella sampling, you're driving it to go 360 degrees, or are you trying to restrict it to certain angles?
So we started, no, we do no restrictions. So I skipped over so many details of the simulation. So thank you for asking. So when so we get our nitrile in, that's just from the crystal structure, right? And then what we do is we position both chi1 and chi2 360 degrees. I think right now we're doing it in 60-degree increments. I think we've decided that that's it gives us the full distribution. Now, some of those structures are going to be sterically prohibited, right? Because it's going to be pointed into another atom or something. And so in the equilibration, those will shake out. But then we'll be left with structures that are chemically reasonable. And that's what we do our umbrella sampling on.
All right. Okay. All right. Okay. Thank you.
Wei.
Lawrence, really nice talk.
Thank you.
And I think I should having Pengyu as your collaborator for the spectrum calculation may be very helpful. I agree.
Yeah.
Otherwise, Pengyu can keep my ass. But anyways, seriously, I think that's good. Right. So the one thing actually I feel satisfactory. I'm satisfied for the fact that you can get a spectrum which agree with the experiment. But I'm still not satisfied for the fact that I still don't learn, right? In a sense, actually, I can have a spectrum, but what do I learn? And
okay.
What this wave tells me, right? It's my naive thought actually. This waves reflect a lot about the dynamics. It's not only about the dynamics near this probe, but even dynamics which are a little bit further away from the probe. Right? And so in that regard, actually, I think actually probably even using amoeba may not be sufficient. Probably you can get a really good shift, right? And you can get a really good absolute number, but I don't know whether you can get a correct distribution. And so this distribution reflects a lot about dynamics, right? And so the particular when you do mutation, then some mutation may not be very close to your probe. And that taught me, the dynamics is very important. Right, and so I can come back to sampling part as well, right? Of course, actually, you were asked about actually the equilibration part of sampling, whether you can get correct, right? But actually, I really worry about that whether the nearby protein response, right? When the probe rotating, undergo the torsional motion, whether the protein nearby, because actually, when you do measurement, there's a different hierarchical time scales motions, whether they're coupled sufficiently. In that regard, 50 nanoseconds is really not enough, right? But the distribution may reflect a really long time scale distribution in a certain way. Right?
Yeah, so the question about what are you actually learning, I think, is really critical, because keep in mind, this is a tool in pursuit of asking difficult questions about what the chemical environment, the structure of a protein, which we can get through many different ways, how that is actually leading to, say, a specific ligand binding or a specific protein binding or a specific affiliation with one part of a membrane versus another, where if we're going to try to reduce that to some sort of quantitative factor instead of just sort of hand waving it all, we're going to need a quantitative measurement like field, right? And so no matter how we're making that measurement, we better understand at least what we're measuring, right? So that's really what this goes to, right? This is in service of making our experimental tool better. And there have been many suggestions now about making the simulation tool better as well. And that's absolutely, I absolutely agree. And so then the question becomes, what can we learn by comparing what we get from simulation and experiment? If we understand exactly what we're measuring, then we can compare that much better to simulation. And as an experimentalist, I would love to be able to do as much as possible in the computer, right, to guide us to new and interesting positions to look at, because these are such large, complex molecules. My poor imagination isn't going to be able to come up with that all, right? So having a calculation that I have confidence in because I've tested against, for example, a data set like this, is also really critical.
Yeah, I think that's really nice. I just feel actually your spectrum really has more information, and then so which allows us to even learn more than something actually about dynamics. I think that's very important, right? And because actually in different part of the protein, you see different the breadth of the distribution, which indicates something really interesting.
Yeah. And so at first blush, like let's take this example right here, this T62, right? It has a very pronounced shoulder here to the red, right? And so you would just look at that without knowing anything more and say, well, there are probably two distinct populations of this nitrile position, one here, one here. Can we find that in the simulation? Absolutely.
Yeah. Thank you.
Thanks, Wei.
Jared Weaver.
Sorry, my computer was taking a second to respond. Thanks for the talk, Lauren. This is it was very clear, and it was interesting to hear about your efforts in trying to make the nitrile IR absorption more interpretable or easier to think about. I was wondering, just on the aspect of, for instance, this slide is a good place. How much have you guys looked at including multiple populations in your IR spectra analysis? For instance, you could imagine a situation where nitrile happens to be in a field and hydrogen bonding orientation such that its frequency would be similar to a nitrile in another, like perhaps non-polar situation. And in that case, having a broad field, a broad band, you might be able to fit it with one Gaussian, for instance, but maybe it'd be more appropriate to choose two. So I'm just curious if you guys have tried multi-population analysis of your FTLS, for instance.
Yeah, good question. We try not to be too aggressive about, for example, taking spectra like these and fitting them. So for example, I just pointed out like this shoulder right here. Okay, we could probably do that safely, right? Not fool ourselves. I think I'm much more interested in when we see something very broad, like these ones here at the bottom. This clearly indicates that our nitrile has a range of environments that it's sampling. Is it spending roughly equal time in them? Or is there one that dominates and one that's in the minority, right? those, again, I think are the types of questions we're going to get best from simulation, if we trust our simulation and we trust our collaborators who help us with that.
All right. Thanks, Lauren. Yeah, that's it. That's good.
Thanks for the question. Yeah.
Okay. We have again a question from Thomas Simonson.
Yeah, Lauren, just one more quick question. You described all these short-range interactions, which are very interesting, and your nitriles typically accept one or perhaps two hydrogen bonds. And so what I'm missing is sort of what is the relative contribution to the local field of these hydrogen bonding groups compared to the effect of the whole protein. I mean, it's like the whole interpretation of the Stark shift methodology depends on, it seems to me, the kind of simple total field being backed out of a vibrational frequency shift. And so how big is the effect of these one or two local groups compared to the total field?
I think that, I mean, you've just asked the single most important question, and the reason we've made such effort to try to deal with the hydrogen bonding part, right? Because if we only have this simplistic interpretation that a shift, is equivalent to a field, that's, on its surface useless, right? And so the environment,
it's a pretty good approximation, actually, and not at all useless, but your work can make all that a lot more precise.
Exactly. And so then the question becomes, okay, you're interested in understanding the field environment in a particular position in a protein. Maybe it's in an active site, maybe it's in an allosteric position, right? That you know about from some other experiment. What happens around that environment? How local does it have to be, right? I mean, on first blush, you might say, well, obviously the hydrogen bonding interaction is going to be the strongest, but no, not obviously, right? So I'm soon to have a new colleague here at UT, Yi Liu, who's moving down from the University of Illinois, and he's looked at changes in redox potentials of metalloenzymes, in which he has made a bunch of mutations. Let's say that the metal's in the active site. He's made a bunch of mutations in the immediate solvent exposure of that metal, right? And he measures changes in redox potential. But then he goes one layer out, and he goes another layer out. And I think he's shown up to 3 sort of shells away from this metal, he can measure substantial changes in redox energy. So, right, the entire environment of the protein is combining at any given position to create the electric field at that environment. And the question that I think we're all trying to ask ourselves is, what part of that is most important? How can we put this together in a mathematical model to get it in a computer, right? And how can we understand how the environment as a whole, even when we make mutations that seem to be fairly far removed from the position we're interested in, affect what's going on at point 0, right? Whether it's the catalytic bond or whether it's a protein-protein binding event or whatever. So I think you've just asked the central question that no one experiment and no one simulation right now, at least, can address. Well, it's this holistic environment.
You asked it, Lauren, not me. That's like all your work is asking that. So, okay, thanks, Lauren.
Anyway, thank you for the question, because it's, I think, the absolute central point of what we're trying to do. Marilyn, you have your hand up again.
Yep.
I do. It's something I'm kind of boring, and I said, is that this is, while this is really interesting, the fields are actually not the things that directly affect, say, pKs or redox energies, because the fields are the gradients and not the potential. And so I think that's probably one of the reasons that you don't correlate with the pK shifts and the fields. and the fields, they're a vector, so they depend on like and we're looking at them for you over a fairly small region, and they're so it's a very detailed, precise measure, but it's interesting to see how that plays out within, say, the way the protein folds up or the way the protein binds things, because it's a different, it's got different properties.
Yeah, no, completely agree, Marilyn. And this is a conversation we've had in many different
Right.
contexts, and I completely agree. And so how does the measurement that I'm making, which again is experimentally convenient, right? That's not necessarily a good reason to do something, but it's what I have. And so how can I interpret that in a real protein, or pick your biological molecule of interest, to understand how a function is occurring, or a protein is folding, or a change in pH is influencing some mysterious function of the enzyme, right? So we have to think about each and every one of those contributions. Nothing is not important.
Yeah, I mean, from seeing your work and Steve's, I mean, you really see that along a bond, when you're doing chemistry, the field's super important. When you look at, say, long-range electron transfer, as we've done, the field through the whole protein can be quite important. The difference in potential at the donor and acceptor site, which is a different scale. So it's just, I don't know. Anyway, thank you.
It's tough. Yeah.
It's interesting.
There is a question from the YouTube chat.
Okay.
From Caroline Fitch. She says, I'm trying to think about global structural dynamical effects on the measurement. Did you have a mutant with the mutation on the surface for a control, and maybe even an unfolded mutant? And finally, in other words, does the slope correlate with stability?
With stability. So first of all, in this particular, I don't have my slides up anymore. In the 10 mutations I showed you, we didn't pick anything explicitly, say, on the surface. We did do a lot of that work with GFP. We always had a control that was completely solvent exposed, measured FTLS and everything, and it was essentially identical to what you would measure in water. And we're working with systems precisely because these are rocks, right? These are very easy proteins to work with. We have to express them in high yield. We do have to do temperature dependent experiments. So we've never seen anything like, say, stability influenced FTLS, but we also haven't looked, right? We haven't done that experiment.
I see. There is a further question from Jacob Kirsch.
Hi, Lauren. Thanks very much for the talk. Very, very interesting. I was wondering, you had one spectra which you had described the frequency was even more polar than water. I was curious how you guys think about that, or might just interpret that.
So I don't have my slides up, and I won't go scrolling to find them. But what that means is that there is a very persistent sigma hydrogen bond, right? So it's blue shifted compared to water even.
And I guess that, if I recall, that distribution, it seemed perhaps even broader than water. So that means that there's a very persistent interaction, but I guess then how do I also kind of think about the distribution in addition to the fact the frequency is so
what that means, and you could go look at structures, but what I would predict is what's going on is, as it's moving, it's taking a hydrogen bond with it. Right? So its chemical environment is changing, but it has either one or several hydrogen bonds that are essentially coming straight on at that lone pair.
Got it. Thank you.
Thank you for all the questions. This has been really enjoyable.
Yes, I Yeah, okay. Let me look, double check again to the YouTube chat. Okay, Caroline is thanking. So let me thank again Lauren for this very interesting talk, and all the people for contributing so lively to this series. I'm very happy, and I thank everybody for making these events such a nice thing. So I will tell everybody, see you next time. I hope the more let's say more interested to the computational side will register to the CCAM event, and for all the rest will meet to the protein electrostatics advances. I'll send an email when they will be ready. Marilyn?
I don't want to leave without thanking, just giving everybody a chance to thank you and say just how amazing this last few months have been, and really how hard it was for you to just everything. So really, thank you, Walter.
It has been hard, but also very rewarding. I've been very happy.
Well, you never know if it's going to, come together, but this one really satisfied an obvious need that we didn't know we had. And so thank you.
Thank you. Thank all of you. Okay. So see you soon. Take care.